and welcome to the Absolute Recap Biology Edition, where we aim to maximize your understanding and minimize your need for memorization. Each episode will recap content, skills, and test-taking tips to help you succeed in May. I'm your host, Melanie Kingett, and your recap starts now. Hi, and welcome to the Absolute Recap Biology Edition. Today's episode will recap crafting a gene. Let's zoom out. We're in Unit 6, Gene Expression and Regulation, topics 6.4 to 6.6. Our big idea is information storage and transmission. Roses are red, violets are blue. If a promoter is blocked, this may be untrue. Unless gene expression is modified, then it could be different. But none of this rhymes. I digress. Today's episode is all about genes, what they are, how they are tweaked, how they turn on and off, and the effect that has on phenotype. Let's zoom in. One of the common mistakes that students make when discussing genes and their expression is using the term gene interchangeably with the term allele. They are related, but not synonymous. A gene is a piece of DNA that carries hereditary information. Wait.
How many years of my life I've spent waiting for you?
Does this mean that part of DNA is not hereditary information? Absolutely. The majority of DNA contains non-coding information. In fact, some estimates claim that as little as 1% of DNA actually codes for functional proteins.
Whoa.
However, research through Stanford University and the ENCODE project, Encyclopedia of DNA Elements, suggests that as much as 80% of DNA has biological functions. No matter which tomato you tomato, there are parts of DNA which are non-coding, and the cell needs strategies to sort out the junk. Okay, back to the difference between genes and alleles. Basically, an allele is an alternate form of a gene that exists in a population. If I go back to our poetic intro, roses are red, violets are blue, and think about Mendel, we can put a few things in context. Genes are located at a specific place on a chromosome known as a locus. And since chromosomes occur in homologous pairs, there are two sets of each gene within a cell. Even if a gene has more than two alleles that exist in a population, like with ABO blood typing, only two can be inherited for any one individual. These copies might be the same, homozygous dominant or recessive, or they might be different, heterozygous. The expression of these alleles also has great variation according to traits that follow Mendel's rules and others that tend to break them. If these flowers were cultivated by Mendel, the genotype might be heterozygous, with a dominant allele capital P and recessive allele little p. Phenotype, purple. Gene expression occurs at many levels, all of which are essential in producing an organism's phenotype. Also, gene regulation is what allows for different cells in multicellular organisms to be, well, different. Eyeballs do not do what elbows do, even though they have the same set of instructions. Eyeballs are reading DNA Manual Chapter 2, while elbows is a short paragraph in Chapter 8. No, the DNA Manual isn't organized into chapters, but rather into chromosomes. But you get the idea.
If you were to find yourself faced with the opportunity to hunt for buried treasure, and you had the option of either A, going on that treasure hunt with an experienced adventurer and access to the treasure map, or 2, winging it and just figure it out as you go. Now, winging it is admittedly very exciting, but the phrase, "I'll figure it out as I go," is usually the last thing I say before getting very lost or spending hours digging through the trash for the furniture assembly instructions I said I didn't need. An experienced adventurer and access to the treasure map is probably the smartest option. Now swap out the word treasure map with study guide, adventurer with teacher, and buried treasure with educational aspirations, and I don't know where I was going with that. But what I do know is that we have study guides, virtual classrooms, and tutoring sessions available now at www.TheApsoluteRecap.com. Now back to the recap.
First, DNA is tightly coiled around histones, forming heterochromatin and euchromatin. Only euchromatin is loose enough to access nucleotides. Second, DNA is double-stranded, but only one strand will act as the template for transcription. Which strand will get this honor? Well, that depends on which gene is being transcribed at the time. Either way, the enzymatic reading occurs 3' to 5'. Since enzymes interact and move unidirectionally, we get phrases like upstream before the gene and downstream within or after the gene. So location matters. Specifically, the location of DNA regulatory sequences that interact with regulatory proteins to control transcription. A short DNA sequence upstream of the transcription start site is known as a promoter. This is where RNA polymerase and other transcription factors bind to initiate transcription before moving downstream towards the gene. Other negative regulatory molecules can inhibit gene expression by binding to DNA and blocking transcription. Genes turn on and genes turn off. Let's say that transcription factors bind to a promoter. RNA polymerase moves downstream and an mRNA transcript is synthesized, 5' to 3'. You still might end up with different protein products. mRNA needs to be processed, with non-coding introns spliced out and exons retained. Known as alternative splicing, different sequences can be removed as a result of different cell signals, resulting in unique transcripts from the same gene. We are going to end today's episode with a recap of the Lac Operon, everyone's favorite bacterial gene expression example. First, the name. Lac, because these genes involve the products which aid in the digestion of the sugar lactose, and Operon, which is a group of genes and regulatory sequences controlled by a single promoter. This is common when genes need to function in unison, when multiple proteins are needed by the cell simultaneously. The Lac Operon is an example of an inducible operon, which turns on only in the presence of lactose. And this makes sense. Why would a bacteria make enzymes to break down lactose if the sugar wasn't around? Both inducible and repressible operons respond to environmental changes, which means better efficiency for the cell. Time for unit connections. Unit 4, the cell cycle. Don't forget that gene expression can be stimulated by signal transduction. For example, cytokines regulate gene expression to allow for cell division, and expression of the SRY gene triggers the male sexual development pathway in animals. Also a connection to Unit 5, heredity, with using probability to analyze single gene inheritance pattern. Additionally, environmental factors can influence gene expression and phenotypic plasticity. Even Unit 7, since cell specialization is a huge factor in survival. All right, what about the exam? Make sure you've got your vocab ducks in a row before diving into practice problems. Genes versus alleles, transcription versus translation, DNA versus RNA, and chromatin versus chromosome, just to name a few. Science is a language first and a concept second. To recap, gene regulation results in gene expression and influences cell products and function. Genes are heritable and vary from one cell type to another. Each gene is transcribed and at times modified before translation and phenotypic expression. Coming up next on the Absolute Recap Biology Edition, helicase unzips. Today's question of the day is about the Lac Operon. In the Lac Operon, is the promoter upstream or downstream from the operator? For the answer to the question of the day, please follow us on Instagram at TheApsoluteRecap. That's the APsolute recap. Check out our website, TheApsoluteRecap.com, for episode schedules, study guides, virtual tutoring, and to sign up for our virtual classroom. The Absolute Recap is produced by Brad Kingett with music by Zach Caruso. Today's episode was written by me, Melanie Kingett. Thanks for subscribing, and don't forget to rate and review wherever you get podcasts. Time's up. Pencils down. Thank you for listening to the Absolute Recap Biology Edition. AP is a registered trademark of the College Board. Copyright 2020, Absolute Recap LLC, all rights reserved.